Good morning. Welcome to Currents Chem World. In this video, let us learn scanning electron microscopy and its principle, instrumentation, and their applications. Okay, before we study SEM, scanning electron microscopy, let us compare electron versus optical microscopy. Okay. Electron microscopes are very similar to optical microscope, but it uses electron beam instead of visible lights. In optical spectroscopy, we use visible light, whereas in electron microscopy, we use electron beam. Okay, second point, it uses electromagnetic lenses, not glass lenses. In optical microscopy, we use glass lenses. In this case, we use electron optics, that is electromagnetic lenses, to control the electron beam and magnify the image. மூணாவது பாயிண்ட் பார்த்தீங்கன்னா, the wavelength of the electron beam is 10 power 5 times smaller than visible light. That is, electron microscope have a much higher resolution of about 0.1 nanometer. Okay, it is ideally suited for nanomaterial characterization studies, etc. Whereas optical above 200 nanometer. Okay, now what are the different types of electron microscopy? There are two main types. One, scanning electron microscopy, it is called SEM, and the second one is the transmission electron microscopy, and another type, scanning transmission electron microscopy. It combines scanning and transmission STEM. It was invented by Ernst Ruska. It was invented by Ernst Ruska. He was a German electrical engineer who invented the electron optics, electromagnetic lenses, and hence electron microscope. He was awarded the Nobel Prize in the year 1986 for physics. Okay, now let us discuss the principle of SEM. SEM gives very high resolution than optical microscope. The electron gun is used instead of visible light and are accelerated to energy in the range 0.1 to 30 kilo electron volt. The energy of the electron beam should be in this range. Okay. It uses a series of electromagnetic lenses to focus the electron beam onto the sample, where it interacts with the sample to a depth of 1 micrometer. The sample depth, it can interact how much? It can interact up to the depth of 1 micrometer. Okay, it scans the surface only. So 1 micrometer depth only. Okay, when electron beam collides on the specimen, it gives two types of signals to generate the image of the sample. They are called secondary electrons and backscattered electrons. 1, 2. Okay. The secondary electrons are obtained from the inelastic scattering, and backscattered electrons obtained from elastic scattering. Okay. Now these are used to generate image. Okay. When a negative voltage is applied to the collector screen, okay, these electrons emitted or collected by collector screen. Okay. When a negative voltage is applied to the collector screen, only the high energy backscattered electron is captured to give the image, because the low energy secondary electrons are repelled by the collector, negatively charged collector plate. Because electrons are also negatively charged, the collector screen is also negatively charged. Okay. Electrons captured by the detector or collector screen, that is photomultiplier tube, amplified to give some image. Okay, now let us see the animation of SEM instrument. As you see, it consists of electron gun. It is a source of electron beam, and it falls on anode plate with the aperture hole at the center. There is a hole through which the electron beam passes through. Why it is used? This is positively charged. The electron beams are accelerated towards the sample by using this. Okay, the electron beam then passes through condenser lens, which defines the size of the electron beam and also the resolution of the image obtained. So it is very important for the resolution of the image, SEM image obtained. And then the electron beam falls on scanning coil, where the electron beams are slightly deflected to fall on here, to here and here, so that the image is obtained from, the signals are obtained from entire surface of the sample. We'll see further the next slide. Okay, and then the electron beam passes through objective lens, okay, which focuses the electron beam onto the sample, okay, to give backscattered electrons. These backscattered electrons are collected and converted into image and displayed here. Okay, let us further discuss the schematic of SEM instrument. It consists of the following essential components we have discussed earlier. It consists of electron gun. It is a source of electron beam accelerated down the column, placed in high vacuum column. All the setup are in vacuum so that electron beam cannot interact with air molecules. And then it consists of electron optics. Electromagnetic lenses include anode plate with aperture to accelerate the electron beam. Condenser lens defines the size of the electron beam and the resolution. Objective lens, it focuses the electron beam onto the sample. Okay, scanning coil is used to deflect the electron beam so that it can scan the entire surface of the sample. Then the backscattered electrons are allowed to fall on photomultiplier to amplify the signal. Computer system is used for image construction, data acquisition, analysis and display. The SEM image is formed from the, for example, okay, if the electron beam hits here, backscattered electrons are obtained. Okay, each spot, the intensity of the signals emitted is analyzed. The SEM image is obtained from the intensity of signals emitted from each spot data point in the sample surface. Okay, let us now see the applications of SEM. It is useful for characterization of surface of solid materials, microorganisms, cells, virus, etc. Basically, SEM provides topographical, shape, morphological, compositional, crystallographic information of materials and cells and microorganisms. SEM can detect and analyze surface non-uniformities, surface fractures, and provide information on microstructure variations, examine surface contaminations also. Further, SEM is useful in determining the spatial variations in chemical compositions. Whether the coating is uniform, it can be obtained from SEM. SEM is spatial variations in coating, chemical composition can be obtained from SEM. And then qualitative chemical analysis and identification of crystalline nature of the nanoparticles or the materials are done by SEM. Whether the nanoparticles are crystalline or powder or grain, etc. And SEM is an important research tool in various fields such as life science, biology, gemology, medical, forensic science, nanoscience, nanotechnology, and in metallurgy. SEM have applicability in several industries including sensor fabrication, battery, solar cells, semiconductor fabrication, inspection and production. Okay, now let us see the SEM image of nanoparticles, where you can see the topographical, shape, morphological features, crystallinity of the nanoparticles. Okay, they are crystalline and have definite shape. And this is the SEM image of red blood cells, which is biconcave in shape. Thank you for watching. Please share and subscribe. Thank you again.